You close your eyes, natural instinct, you don't have to think about it, right? So that no more entry of foreign things, and then tears come out. It's going to wash the eye. What does tear contain? Is it just water? No.
It's solution.
It's a solution of things. It has lysozyme, antimicrobial enzymes. The eye is trying to protect itself, right? So now in some people, the gene that codes for this enzyme, the gene that codes for this protein becomes faulty, defective. So that means their body cannot make any more lysozyme. That means now they have less immunity, less protection. So scientists have come up with this. This goat is a transgenic, carries a gene for an enzyme called lysozyme. So then what will happen is this goat will make a lot of lysozyme enzyme. That lysozyme enzyme can be extracted from this goat's whatever body fluids, right? And then that can be packaged, bottled, can be used as a medicine to supply, to give to those people who are not making lysozyme enzyme. So this is all genetic engineering, recombinant DNA technology. These are things that are being commonly used, or a protein which helps dissolve blood clots. Otherwise, this could be fatal. It could kill a person. Okay. Vaccines. Nowadays, we come up with vaccine. How did the COVID vaccine came up so fast and saved millions of lives? Because of recombinant DNA technology. We already had the mRNA of COVID, similar to COVID virus. Scientists had that mRNA sequence. Erythropoietin, EPO, this is used to treat anemia, people who do not make enough hemoglobin, okay? Erythropoietin it helps people to make more red blood cells. The more red blood cells you have, the more hemoglobin you have. The more hemoglobin you have, the better the respiration, the better the oxygen transport within the body. So that is all being done by the recombinant DNA technology. Genetically modified organisms. You see here, breeding programs, most of the corn, soybean, cotton crops are genetically modified. Now I know there's a lot of ethical questions that come out. Are these really healthy? Well, we need, of course, 20 more years of research to really come up with long-term effects. At least short-term effects, not that much. And the problem is, where is the solution? There is no other option. If we do not use genetic engineering, if we do not come up with genetically modified organisms, how can we feed our increasing human population? We cannot. Using normal traditional ways of agriculture, that's not enough. Human population has just, we have become like the largest population now, using the maximum resources from earth. So we have to use genetically modified organisms. Why? Because then they can be made insect resistant, they can have higher yield, or for example, an example has been given for strawberry plants, where a bacterial gene has been introduced that acts as an anti-freeze, so that when it gets too cold, if it's not a genetically modified strawberry plant, that plant will freeze and die. No more fruits coming from there. But because of this, now these plants can survive freezing temperatures. Or researchers are trying to come up with an edible vaccine against cholera. Cholera is a big killer, not in US, but in a lot of other countries around the world. So, and a lot of children die from cholera. And when it's an injectable form, then kids are scared, and also it's more difficult to transport and all. But if we can come up with an edible form, like those Tums tablets that you eat, easy to transport, easy to give to children, so easy to save life. So that's why if we can come up with an edible vaccine against cholera, that will save lots of lives. This is a variety, a transgenic variety of rice that contains genes from these flowers, daffodils, and also corn. This rice, why are these genes introduced? Because these can prevent vitamin A deficiency, which causes blindness in people. So there are parts of the world where people are highly deficient in vitamin A. If you can give them this rice, then they don't have to take the vitamin supplement separately. They are getting it. Just like we don't have to take iodine supplement separately. Why? Because our salt comes reinforced with iodine, iodized salt, right? So that was the government initiative. Or cassava, which is a starchy root that's a staple for a lot of people, Africans, continent Asia. This has been modified to produce increased levels of iron and beta-carotene, which is converted to vitamin A in the body. So you see, a lot of these genetically modified organisms or crops have been created to help people fight deficiencies and diseases. So genetic engineers are targeting not only plants but also animals. For example, for cattle, a gene that causes development of larger muscles, right? So that is being used to make chickens and cattle larger, or genetically modified pigs which carry a roundworm gene whose protein converts less healthy fatty acids to omega-3 fatty acids. Have you guys heard of omega-3 fatty acids? Can you give me good sources of omega-3 fatty acids?
Salmon.
Salmon. What about for vegetarians? Nuts, chia seeds, quinoa. So those are all good sources of omega-3. Now omega-3 actually, there is a lab in Loma Linda, and I also collaborate with them. They have done experiments on rats, and one of my former students, anatomy students, he is getting into medical school. He wanted to get some research experience, so I sent him a letter. I gave him a recommendation letter. He was doing research at Loma Linda and on the same things, and what they do is they take rats, a group of rats, they will give omega-3 fatty acids, okay? The other group, they will not give them omega-3 fatty acids, and they induce spinal cord injury in both groups. And then they check for which group of rats recover faster, or which group of rats has less neuronal damage. So they always find that the group that receives omega-3 fatty acids, they recover faster and they also have less damage, right? So that means if you are already taking omega-3 fatty acids, if you are already eating salmon or chia seeds or whatever nuts, then in case there is an accident, something, then the probability or the chances of nervous system damage are less in those people. Another example of genetically modified organism is this Atlantic salmon that has been genetically modified to become big, reach market size in half the normal time, 18 months versus 3 years, to grow twice as large. Okay. Human gene therapy.
Hi, I'm Jean, and I'm here to help you understand what gene therapy is. But first, we need to learn about genes. Genes are the blueprint or instruction manual of all living things and can be found in each and every one of your cells. From this blueprint, cells are able to get to work. Genes affect everything from the color and texture of your hair to your height to how your body controls bleeding. Genes are made from your DNA and carry your genetic information. Sometimes there's a change, aka a mutation, in the gene from the original copy. This mutation or defect in a gene causes it to not work properly. Genes that aren't working properly can have serious consequences. Scientists are working to find ways to solve this problem. They can replace, add, or turn on or off genes to tackle many health issues. Mutations can come from biochemical processes within the cell, something outside of the cell, or what we inherited from our parents. In hemophilia, when a mutation happens, it causes the body to not be able to produce clotting factor. Disorders like hemophilia, VWD, and other types of bleeding disorders are mostly from inherited gene mutations. When a mutation happens in hemophilia, the blood doesn't clot properly, and this is where gene therapy comes in. Gene therapy is the introduction, removal, or change in genetic material. For hemophilia, it's the introduction of a modified gene. A functional gene is made outside the body in a lab, and then it is transferred into the body. In the lab, scientists can repair a defective gene. Sometimes they add a new gene to fix a problem or even turn off a problem gene. In hemophilia, it's the introduction of a new working gene. Once the gene is modified, it changes how a protein or a group of proteins is made, meaning it changes how the cell works. In the case of hemophilia, this means the cell makes new proteins that allow the blood to clot. But to get into the cell, the new gene needs to first hitch a ride. Scientists use viruses. These viruses are called vectors, which are like little delivery trucks to get the repaired gene into your body. Scientists make a virus safe by removing all the stuff that can make you sick. Then, the delivery truck, aka vector, can carry the repaired gene into the cell, and your body can make the clotting factor that helps your blood to clot. Gene therapy is bringing us closer to a reality where we have new options to treat rare disorders like hemophilia.
So this was gene therapy explained very simply for you to understand. There is a more text version of that given how it is used. So now you know that in gene therapy or genetic engineering we use both bacteria and viruses, but of course bacteria and viruses are first made risk-free, and that is when they are used, right? Scientists will, or medical companies, the biotech companies will not use them if they are still harmful or can cause a disease. So in 2009, an example, researchers, they found a particular kind of blindness which was progressive, means gradually increasing. This was linked to a defective gene, and the gene was responsible for making pigments that could detect light in the eye. They found that if they gave the patient a viral injection, an injection containing the viral vectors, and those viral vectors contained the normal gene from human beings. So if they introduce those viral vectors containing the normal human gene into these people, into these patients who were suffering from the disease, this injections actually helped a lot. Okay, they restored their vision to a large extent without side effects. Two other illnesses that have already been treated with gene therapy, okay, in few patients, are a degenerative disease of the nervous system and a blood disorder using hemoglobin gene, for example, hemophilia and neurodegenerative disorder. The main problem is, of course, our nervous system disorders. The reason being, neurons do not undergo mitosis. So, natural replacement and recovery is almost zero. And that is why we need these kind of treatments for patients who are suffering from neural problems, schizophrenia, Alzheimer's, Parkinson's, dementia, or an injury due to an accident that causes the patients to be wheelchair bound or bedridden, right? So we want to help these patients, and these are our only options. Now we move to DNA profiling and forensic science. So how is DNA profiling done? Let's say some blood was obtained from the crime scene and we have two suspects, there was a murder. So we take the blood, we take the DNA out, DNA extraction, we will amplify it using PCR, and then we do a gel electrophoresis to see the DNA bands. Okay, this is the gel which the DNA band shows. So from the crime scene, the blood gave out these DNA bands. You see a pattern of bands here, right? And now look at we have suspect 1 and suspect 2. We took their blood samples. We did the same thing, DNA isolation, PCR and gel electrophoresis, and we got their banding patterns as well. So now you have 3 band patterns. This is crime scene band pattern, suspect band pattern, and suspect 1 band pattern, suspect 2. Which is the matching one? 2, suspect 2's DNA pattern, gel pattern matches with that of the crime scene. So now the police can easily file charges against this particular suspect 2. Now a real life example, a long time ago when DNA analysis was not available or was not accepted as correct evidence, there were people who were wrongly imprisoned and they spent many years in prison. This is one such gentleman there. So there was a rape case, there was semen on victim was found, and because at that time these more modified sophisticated analysis is called as STR or short tandem repeat analysis was not available. So based on somebody saw someone and whatever, it was dark, but I think this was this person, so this particular gentleman was convicted and he served for many years in prison. It was only much later when DNA analysis became admissible in the court of law and was accepted, and that's when his case was reopened, like many others whose cases were reopened. And when again this DNA analysis was done, Earl Washington, his STR sequence did not match with the semen that was found in the rape in the crime scene. On the other hand, there was this other guy who they did not suspect, his was a perfect match. You see the STR sequence match? So, and you might have probably heard of these things, nowadays there are groups of people who help these kind of prisoners who have been in the prison. There was no DNA analysis done and they still claim that they were innocent, but because of whatever evidence, circumstantial evidence was present, they have been imprisoned for many, many years. Now we move to some of these techniques. Remember I said when DNA is extracted, how do we make large amounts of DNA? It's called the polymerase chain reaction, PCR. So we take a little bit of DNA, and this is how much DNA we are getting after every cycle of amplification.
We're investigating a crime scene and only find a tiny drop of blood or a hair follicle as clues. Although these pieces of evidence have DNA, they only have a very small amount, not nearly enough to deduce who they belong to. So how can you get enough DNA from this evidence to help solve the crime? The answer is PCR, or polymerase chain reaction. PCR is an important biotechnology technique that allows us to make many copies of DNA. Essentially, PCR is DNA replication in a test tube, and it allows us to get sufficient DNA to determine which suspect was present at the crime scene. Here's how PCR works. A special microfuge tube called a PCR tube is filled with all the stuff that you will need to copy the DNA of interest. This includes 1. The crime scene DNA you wish to copy. 2. DNA nucleotides. These are the building blocks of DNA that will be used to build our copies. 3. Primers. These are needed to get the process started. 4. A special form of DNA polymerase that tolerates high temperatures, known as Taq polymerase. Taq polymerase comes from a bacterium that lives in hot springs with nearly boiling water. Since PCR uses high temperatures, this type of DNA polymerase is necessary. Other types of polymerase would denature at these high temperatures. Once all of these reagents are added, the PCR tube is placed in a PCR machine, also known as a thermal cycler. Inside the PCR machine, the DNA is heated, breaking the hydrogen bonds that hold its two strands together. The machine then cools down the reaction mixture. This allows primers to attach to the ends of the DNA segment that we want to copy, which is usually a region that varies a lot between different individuals. Now that the primers are attached, we can use Taq polymerase and nucleotides to build new DNA strands. The PCR machine raises the temperature to the optimal temperature for Taq polymerase, so that this enzyme can get to work. Nucleotides are added following base pairing rules, just like in normal DNA replication in a cell. This process of heating and cooling the PCR mixture is repeated many times, and every time it is repeated, the amount of DNA doubles. This can make a lot of DNA. After 30 cycles, for example, a single molecule of DNA can become over 1 billion copies. This amount of DNA is plenty to be able to analyze using other techniques like gel electrophoresis, so that we can determine whose DNA it is. If you enjoyed this video, please like it, subscribe, and check out the other free game.
So this was PCR. It is routinely used in any molecular biology lab. When I was doing my PhD in India, that was one of the best research institutes in India, but we had only 2 PCR machines, right? So all PhD research scholars, we would have to book long time ago. Like I am getting a slot available in 2 days. And all the reagents that you need to start this recipe, you saw primers, oligonucleotides and Taq polymers and all. So we had to order, the best quality came from US. So we would order to a company called Sigma, and they would deliver it like maybe 3 times in a year. That means we would wait the entire year to get the supplies, and then as soon as we got it, we didn't have time to waste. We would just have to do. And there was no scope for errors. You miss something, you wait for another 3 months to get the research supplies, right? So the way I was trained in research was to do meticulous planning and then execute it like perfectly, because your scope for error is like 0.001%. You lose 3 months of your time, And then I came for my first job in US. I was interviewed at University of California, Berkeley. That's like one of the premier institutes in the country, the entire world, right? So many Nobel laureates and all. So I came here. And I went and I was told my project was working on lung cancer from patients in arsenic exposed, and I have to work on thousands of patients. And I was like, how long it's going to take me if I have to work on thousands of patients? And then I went to the lab, big lab, huge lab. The mentor is a world-renowned person. And I saw, like series of PCR machines lined there. And I was like, oh, it's like a paradise, dream for a research scientist. I don't have to wait for one machine at a time, because one machine can only run 96 samples. If I have to do thousands of samples, I better have a lot of And it's like you book it and you get it the same day. It's your own PCR machine. So most of the times I would be, starting the experiments in the evening, turn them on, the PCR machine goes on for hours. I come back at night, I take my samples out, I start doing my next analysis. And what people in the lab were very impressed, and they would show my experimental results, is how perfectly she does the experiment. And probably they didn't understand how we were trained in India, where you do not have resources. So you really need to be perfect in doing things in order for you to not waste resources. There isn't money for those resources, right? So here we are, because we get so much in this country, we are kind of, most of us, all of us, we waste a lot. You go to other countries, not as many resources, you learn to conserve, you learn to save, you don't waste. Have you seen all those lights on in stores after they're closed? All the lights on? That's such a wastage of power, electricity, right? So I had to do a PCR on these lung cancer samples and controls for thousands of patients from Argentina, lung cancer patients, and we were doing, and so just a small, I told a small story so that you understand that this is very commonly done in all labs. So then, after you get the DNA, these are some of the things that you get after you have gotten the DNA, some of the techniques that are used for forensics or other experiments, like, remember the STR that I was talking, which was used to exonerate the Earl Washington, who was not actually the perpetrator. So these are short sequences of DNA repeated many times. So we use STR analysis for DNA profiling. Repetitive DNA, we all have repetitive DNA in our sequence. Those are our unique landmarks. So for example, my STR sequence might be in one particular place, I have T, 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 T. Well, another human being, your STR might be different, right? Another student, their STR will be different. So my STR is my own unique landmark. For others, it would be different, and those are used to identify. So short tandem repeat. So imagine, so you see here, let's say this is crime scene DNA, this is suspect's DNA. So now they are trying to identify the STR analysis. So they have taken two STR sites, STR site 1, STR site 2. So between the crime scene and the suspect's DNA, STR site 1 is giving the exact same repeats. But look at the STR site 2, this is different. STR site 2 for the suspect does not match with the STR site from the crime scene. That means this suspect has not done or has not committed the crime. He is not the criminal because the STRs don't match. So how do we look at these STRs? How do we look at DNA? We have extracted DNA, we have amplified. How do we get those bands? Those bands are obtained by this method called gel electrophoresis, which is a method for sorting macromolecules. We can use a gel to get protein bands or to get DNA bands. And it uses the electrical ability of the DNA. DNA is negatively charged. So if you subject DNA to an electrical field, have you heard of this term? Opposites attract. So you know there's an electrical field that's working, right? When the earth has magnetic fields. So because DNA is negatively charged, if you put DNA in a solution where there's a positive charge and a negative charge from one side, DNA will always migrate to the positive side because DNA is negatively charged. So it is getting attracted by the opposite end. And that is the main principle that is used to do gel electrophoresis.
So kind of like a magnet.
Yes.
Once DNA has been extracted from a sample and a section bearing useful code such as a VNTR or STR is isolated, the next step in DNA fingerprinting is to measure each strand and count the number of repeated sections. To do this, scientists use a technique called gel electrophoresis, which uses an electric current to push strands of DNA through a slab of gel-like material. Because each bit of DNA is negatively charged and subject to an equal electric force propelling it to the positively charged side of the gel, smaller pieces move faster than larger ones. When the current is removed, the gel is photographed to reveal how far each bit has migrated. By comparing the bands produced by the DNA samples of interest with bands produced by standard samples whose sizes are already known, the precise length of each DNA fragment can be gauged.
So this is how basic gel electrophoresis is done. DNA sample is taken, it is broken into small fragments, then it is put in a gel, and gel is subject to an electric current. DNA will start moving towards the positive side of the the positive electrode. And because DNA fragments are different size, the ones that are smaller sized fragments, they travel faster. The ones that are larger size, they travel slower. So the larger DNA fragments stay, because they're traveling slower, they travel to the top of the gel, while the shorter ones have traveled to the bottom of the gel. So if you see any gel electrophoresis results, if you see anything like this, for example, this is schematic representation of a gel electrophoresis. That means a band here is a bigger size band than a band here, because this is where the DNA was injected, and this is how the bands migrated. So this one will be a shorter band, this one is a lighter band, this one is a heavier band. But I have at least obtained a band pattern. And when I subject the DNA of the suspect 1, 2 and 3, expose them to the same conditions, their DNA will also, the DNA strands will also separate like this, DNA fragments will separate like this, and we will get a banding pattern. These are called banding patterns. And the banding pattern shows similarity or differences between the crime scene and the suspect. So this is an actual gel, what an actual gel looks like. This is just a representation. You see a mixture of DNA is introduced into, these are called wells of the gel-like material. Then you subject them to electric current, and this is what the completed gel will look like. Now for FBI in US, it has created what is called the CODIS. CODIS means Combined DNA Index System. Combined DNA Index System uses 13 short tandem repeats. So you see here all the chromosomes are taken. Chromosome 1 through 22 are autosomal chromosomes, chromosome X and Y, and they have identified certain STRs, okay, short tandem repeats. So whenever they get a suspect DNA, a suspect blood, and they want to match it with the blood from the crime scene, they simply compare the STRs, all these 13 STRs, okay? And all these 13, so the probability that any two individuals will have all the 13 markers is 1 in 250 trillion. That's impossible. And that is why this evidence is now admissible in the court of law. Because when evidence shows CODIS match, okay, the blood from the crime scene has the pattern of these 13 markers was like this, and the blood from the suspect, the pattern matches in all the 13 STRs. When there's a perfect 13 STR match, there is no way that there is a mistake in the evidence. So this is molecular level evidence. So, so DNA profiling has been used extensively. 2001, September 11, right? World Trade Center. So the bodies were beyond identification. We just, were able to obtain just remains. How do you give them back to the family? You have to do identification by DNA. So you get DNA from the family, cousin, whatever group, and then you try to identify, match it with the DNA from whatever remains have been obtained. So DNA profiling was used. What else? Sally Hemings, okay? It was a DNA profiling that showed that Thomas Jefferson had fathered a child with Sally Hemings, right? So all the descendants, and now you probably, if you Google, you find Sally Hemings' side of the family having a reunion with Thomas Jefferson's wife's side of the family. So that evidence now has been proved by the DNA profiling technique. Or after French Revolution, when the French royalty, everybody was killed, there were rumors that one of the descendants of the French royalty had survived. Many people came forward, or even in Russia, Anastasia, when so many women have come forward claiming that they are Princess Anastasia, and then DNA analysis is done, and of course it's found that they're all fake. So we are done, and we'll continue with bioinformatics in the next lecture on Thursday.